Yeah, good morning, and you're welcome back to the channel. It's always beautiful having you here. Today, we're just going to spend a little time discussing selective and differential media. Now, it's important to note that in microbiology, the core part of what we do is the culture of microorganisms. We want to grow microorganisms from different types of samples, from water samples, clinical samples, environmental samples, from the human body, human waste, all sorts of samples are there, and we want to grow our microorganisms out of these samples. Now, to be able to grow our microorganisms out of these samples, there are different, culture media that we use. And culture media, they're important because different types of culture media, provide different conditions for growth and provide different conditions that allow the growth of different organisms. So this means that culture media can be favorable to a particular set of organisms and not so favorable to another set. They create different conditions for growth. Now, this is important because for today, we just selectively talk about two types of culture media, and that's the what we call the selective media and the differential media. These are two different types of media. Now, when you're talking about selective media, you're talking about culture media that encourages the growth of particular organisms at the detriment of another set of organisms. So if you have two sets of organisms that have some characteristics that define them, that make them different, you may decide to grow a particular set of organisms at the detriment of another set. To be able to do that, you effectively you might want to use different kinds of selective media. A simple example is a sabrowed agar. This sabrowed agar now, or even our potato dextrose agar, these are media that encourage the growth of fungi. So you add your antibiotics to this media, and then they encourage the growth of fungi while discouraging the growth of other bacteria. So this kind of media, you refer to them as selective media because they're encouraging the growth of one organism, and they are doing that at the detriment of another set of organisms. Now, it's important for us to understand that there are different types of mechanisms which this media used for their activity. Some of these media use antibiotic activity, meaning that they have antibiotics being added to them in order to help them for whatever function it is that they want to do. That's selective media that we're talking about. Some other ones use the presence of dyes. They have particular dyes, DYES, particular dyes that help them. So those dyes can encourage the growth of some organisms to the detriment of other type of organisms. Some other media, an example being our mannitol salt agar, have the presence of high concentration of salts. And this encourages the growth of organisms we refer to as halophiles, which are organisms that can grow under high salt conditions. So, an example being our staphylococcus. So, you have mannitol salt agar being a selective agar and helping for the growth of particular organisms to the detriment of other types of organisms. So, it helps organisms that are able to grow in high salt environment. We also have some like our maconke agar. It's also an example of a selective agar, and within it is the presence of biosalts. And these biosalts are can be used by some organisms for their growth and are harmful or cannot be used by other organisms for their growth. Now, there are many different types of selective media, but we'll just mention a few of them. Like I said before, your mannitol salt agar is there, your maconke agar is there, your sabrowed dextrose agar is there, your Thea Martins agar is another example. So they are all different types of selective media. Your mannitol salt agar is there and it helps for the growth of our staphylococcus. We also have our maconke agar, which is there to encourage the growth of enteric bacteria. Many of these enteric bacteria also use lactose for as a source of sugar. You have your sabrowed agar, your potato dextrose agar. They are there to encourage the growth of fungi. You also have your thiametins agar, which is there and it inhibits the growth of many organisms except for your Neisseria. So if you suspect that somebody has a sexually transmitted infection in the form of Neisseria gonorrhea, you can use your Martin's agar to culture your samples and see whether you're able to grow out the Neisseria organism from this agar. These are important to know. Then your differential media. What's the differential media? Differential media is the media that helps to indicate, it shows that a particular set of organisms are growing on that media and is able to show that using dyes and color change. Now look at the difference between your selective and your differential. Your selective is actively encouraging the growth of one at the detriment of the other, while your differential media is just there to differentiate, to show by means of a color change that a particular organism is growing on this media as opposed to another kind of organism. So it visually shows us, usually through color change, that a particular type of organism is growing there. And what are some of the mechanisms by which differentiation occurs? Sometimes we have the incorporation of specific carbohydrates, like our mannitol, like our sucrose, like our lactose. Specific carbohydrates can be incorporated into this agar and making the agar be different. Sometimes too, we use chromogenic substrates. Chromogenic, we know when you hear chromogenic, what comes to your mind is a change in color. So sometimes we have to use substrates that bring about a change in color, and this can be seen in different media. Then sometimes some media have precipitative salts. So the presence of the growth of particular organisms can elicit precipitation of some salts from that agar to show that those organisms are present. So these can be seen in some kinds of agars. Now, examples of our differential agar are like our blood agar, like our chocolate agar that we use for hemolysis. You know that once you're seeing those changing colors of the agar from red to light yellow to lighter, and all those color changes indicate to you that the organism that is growing on that agar is an organism that causes hemolysis. So your blood agar, your chocolate agar are differential media. Our mannitol salt agar, our eosin methylene blue (EMB) agar, and our xylose lysine XLD agar are all examples of our differential agar. Now it's important to note that sometimes there's an overlap between selective and differential media, meaning that some media are selective in nature, meaning that they encourage the growth of particular organisms. Then dyes are actually are also added to this media, to show visually that those organisms that they are encouraging to grow are growing, making such a media being a mixture of both selective and differential media, like our conke, our mannitol salt agar, our EMB. These are all examples of media that you have both types of media growing here. Now, what's the application of all of these kinds of media? The applications are simple. You have applications for them in clinical medicine, when somebody is suspected to be suffering from a disease or a particular outcome, we want to be able to analyze the samples from such an individual: saliva, feces, urine, blood samples, whatever samples they are, biopsies. We analyze them, try to grow microorganisms out of them, and then we have this selective or differential media. We use them to be able to identify these organisms or identify direction in which these organisms are growing. You also have application in environmental samples. If we are doing environmental research, if we are doing environmental studies, we can take samples and then we use this media to grow out our organisms. We also have an industrial application, food and water microbiology, all these aspects of microbiology. We use all these different types of media. So thank you so much for listening to this. I hope you've gotten an understanding, or at least some basics about differential media and selective media. Please remember to like us, share our content, ask us questions, engage with us. And we are here to grow this channel as much as we want it to be. So do have a wonderful day. Thank you for listening and have a very wonderful day.